Hello friends, today I'm here with you with my favorite topic, that is flow cytometry.
And
And
actually,
actually
in
in
my
my
research,
research,
I am
I'm
doing
doing
flow
flow cytometry
cytometry from
from
last
last
9
9
years,
years,
and
and
it
it
is
is
very
very
fascinating
fascinating
to
to
see
see
that
that
how
how
flow
flow
cytometry
cytometry
based
based
analysis
analysis
has
has
developed
developed
in
in
past
past
10
10
years
years
or
or
past
past
15
15
years.
years.
It's
It's
an
an
amazing
amazing
and a
and a
very
very
important
important
technique
technique
of
of
cell
cell
biology,
biology,
immunology,
immunology,
molecular
molecular
biology.
biology.
And
And
I
I
always
always
say,
say,
when
when
people
people
ask
ask
that
that,
what
what
is
is
the
the
limitation
limitation
of
of
flow
flow
cytometry,
cytometry?
or what
What
is
is
the
the
application
application
limitation
limitation
of
of
flow
flow
cytometry,
cytometry?
then
Then
my
my
answer
answer
is
is
always
always
imagination
imagination
is
is
your
your
limitation.
limitation.
So
So
today
today
we
we
are
are
going
going
to
to
discuss
discuss
flow
flow
cytometry
cytometry
based
based
analysis,
analysis,
and
and
it
it
will
will
be
be
a
a
very
very
easy
easy
guide
guide
for
for
students
students
because
because
whenever
whenever
students
students
try
try
to
to
understand
understand
about
about
flow
flow
cytometry,
cytometry,
they
they
miss
miss
the
the
point
point
because
because
there
there
is
is
too
too
much
much
of
of
electronics,
electronics,
too
too
much
much
of
of
mathematics,
mathematics,
too
too
much
much
of
of
physics.
physics.
But
But
today
today
I
I
will
will
be
be
explaining
explaining
the
the
basic
basic
principle
principle
of
of
flow
flow
cytometry
cytometry
in
in
a
a
very
very
simple
simple
manner,
manner,
in a
in a
step-by-step
step-by-step
manner,
manner,
and
and
I
I
will
will
be
be
talking
talking
about
about
some
some
very
very
interesting
interesting
applications
applications
and
and
a
a
very
very
small
small
intro
intro
to
to
quantum
quantum
dots
dots
and
and
their
their
possible
possible
use
use
in
in
flow
flow
cytometry.
cytometry.
So
So
let's
let's
start,
start,
guys.
guys.
So
So
friends,
friends,
first
first
of
of
all,
all,
we
we
understand
understand
the
the
meaning
meaning
of
of
word
word
cytometry.
cytometry.
Cytometry
Cytometry
is
is
a
a
technique
technique
where
where
we
we
measure
measure
physical
physical
and
and
chemical
chemical
properties
properties
of a
of a
single
single
cell.
cell.
And
And
if
if
I
I
include
include
a
a
fluid
fluid
or
or
fluid
fluid
stream,
stream,
then
then
it
it
becomes
becomes
flow
flow
cytometry.
cytometry.
Flow
Flow
cytometry
cytometry
is
is
nothing
nothing
but
but
measuring
measuring
physical
physical
and
and
chemical
chemical
properties
properties
of
of
a
a
single
single
cell
cell
in a
in a
fluid
fluid
stream
stream
in an
in an
instrument
instrument
which is
which is
called
called
as
as
flow
flow
cytometer.
cytometer.
So,
So,
in a
in a
fluid
fluid
stream, if
stream,
we
if
are
we are
doing
doing
cytometry,
cytometry,
then
then
it
it
is
is
called
called
as
as
flow
flow
cytometry.
cytometry.
Now, we
Now we
will
will
be
be
discussing
discussing
about
about
simple
simple
steps
steps
of
of
the
the
flow
flow
cytometric
cytometric
analysis,
analysis.
then we
Then
will
we
be
will be
going
going
towards
towards
the
the
applications.
applications.
To
To
do
do
the
the
flow
flow
cytometric
cytometric
analysis,
analysis,
we
we
can
can
start
start
from a
from a
tissue,
tissue,
or
or
we
we
can
can
start
start
from
from
single
single
cells,
cells,
or we
or we
can
can
take
take
any
any
other
other
source of
source of
cells
cells
like
like
culture
culture
cells,
cells,
cell
cell
lines,
lines,
and
and
bacterial
bacterial
cultures,
cultures,
etc.
etc.
Whatever
Whatever
source
source
you
you
are
are
starting
starting
from,
from,
you
you
always
always
have
have
to
to
prepare
prepare
a
a
cell
cell
suspension,
suspension,
and
and
this
this
cell
cell
suspension
suspension
will
will
be
be
used
used
as a
as a
sample.
sample.
Now
Now
you
you
put
put
that
that
cell
cell
suspension
suspension
in
in
flow
flow
cytometric
cytometric
cuvette,
cuvette,
and you
and you
put
put
this
this
cuvette
cuvette
in
in
the
the
flow
flow
cytometry.
cytometry.
Now,
Now,
flow
flow
cytometer.
cytometer.
Now
Now
the
the
flow
flow
cytometer
cytometer
will
will
suck
suck
up
up
your
your
sample
sample
and will
and will
start
start
analyzing
analyzing
it.
it.
Now we
Now we
see
see
what
what
happens
happens
inside
inside
a
a
flow
flow
cytometer.
cytometer.
If we
If we
talk
talk
about
about
the
the
functionality
functionality
of
of
an
an
instrument,
instrument,
then
then
what
what
happens?
happens?
Your
Your
sample
sample
is
is
going
going
in
in
form
form
of
of
cell suspension
suspension
where
where
you
you
are
are
having
having
so
so
many
many
cells.
cells.
Now
Now
this
this
sample is
sample is
squeezed
squeezed
by
by
a
a
flow
flow
of
of
fluid,
fluid,
and
and
this
the
squeezing
squeezing
of
of
sample
sample
is
is
called
called
as
as
hydrodynamic
hydrodynamic
focusing.
focusing.
Nowadays
Nowadays,
there
there
are
are
some
some
instruments
instruments
which
which
are
are
using
using
both
both
the
the
hydrodynamic
hydrodynamic
pressure
pressure
as
as
well
well
as
as
the
the
acoustic
acoustic
waves
waves
to
to
do
do
the
the
focusing
focusing
of
of
cell.
cell.
What do
What
you
do you
mean
mean
by
by
focusing?
focusing?
Once you
Once
are
you are
applying
applying
this
this
hydrodynamic
hydrodynamic
pressure
pressure
on the
on the
cells,
cells,
then
then
cells
cells
are
are
coming
coming
in the
in the
machine
machine
as a
as a
single
single
array.
array.
It
It
means
means
the flow
flow
cytometer
cytometer
principally doing
principally
nothing
doing
but
nothing but
creating a
creating a
condition
condition
where
where
you are
you are
having
having
one
one
cell
cell
at a
at a
time
time
passing
passing
through a
through a
stage
stage
where the
where the
laser
laser
is
is
interacting
interacting
with
with
that
that
cell.
cell.
So
So
again,
again,
you
you
are
are
having
having
your
your
cells
cells
in
in
suspension.
suspension.
You
You
have
have
created a
created a
hydrodynamic
hydrodynamic
focusing, that
focusing,
is
that
the
is the
function
function
of
of
the
the
machine,
machine.
and
And
because
because
of
of
that
that
hydrodynamic
hydrodynamic
focusing,
focusing,
we
we
are
are
having a
having a
condition
condition
where
where
cells
cells
are
are
present
present
in
in
single
single
array.
array.
Now,
Now,
these
these
single
single
cells
cells
will
will
reach
reach
to a stage where on my left side I am having a laser. The laser will put some light on this cell. The cell will interact with this light, and this interaction is detected by different detectors. So till now, in the first step of principle, what we have learned that flow cytometry is doing nothing but creating a single array of cells which are interacting with the laser one by one. Now we see what happens with the cell when it is interacting with the light. So here is our cell and here is our laser. The laser is putting an incident light beam on the cell, and cell is doing different kind of interaction with this light. The first kind of interaction is the scattering of light at small angles. So this scattering of light at small angle is between 0.5 degrees to 5 degrees, and this scattering of light is called as forward scatter. That is the first parameter of flow cytometric analysis. So what is forward scatter? Forward scatter is scattering of light by the cells at small angle, which are 0.5 degrees to 5 degrees. Now, which kind of information we are getting from forward scatter? Forward scatter is giving us the information about size of the cell. So first interaction is scattering of light at small angle between 0.5 degrees to 5 degrees, which is called as forward scatter, and is giving us information about the size of cell. Now, the second interaction of the light is scattering of light but at larger angles of 15 degree to 150 degree. So this scattering of light at larger angles between 15 degrees to 150 degrees is called as side scatter, SSC, which is the second parameter of the flow cytometric analysis. Now again, the second type of interaction is scattering of light at 15 degrees to 150 degree, which is called as side scatter, SSC. And which information we are going to get from the side scatter? Side scatter is going to give us information about internal granularity of cell. So if we are having bigger size, then we get different type of forward scatter, and if we are having different kind of granularities inside the cell, then we are having different values of side scatter. So forward scatter and side scatter are two basic parameters of flow cytometric analysis. Forward scatter is giving us information about size of cell, and side scatter is giving us now if the cell is having fluorescent factors or fluorescent proteins or fluorescently labeled antibodies which are bound with specific protein, then third type of interaction will come. These fluorescent factors will absorb the light and emit the fluorescence, and this emitted fluorescence will be detected by different detectors, and these third kind of interactions will be giving us information about these fluorescent molecules. So three interaction: FSC about size, SSC is about granularity, and fluorescence about presence or absence of fluorescent proteins or fluorescent molecules on the cell or inside the cell. So the flow cytometer is working with only two basic principles. One, creating hydrodynamic pressure focusing, where we are getting single cell at a time, and second, the single cell is interacting with the laser or incident light beam in three different manners: FSC, SSC, and fluorescence. So again, I am summarizing here that forward scatter is giving us the information about size of cell, side scatter is giving us the information about internal granularity of cell, and fluorescent signals can be inherent fluorescent proteins, then fluorescent proteins expressed with the help of genetic engineering. Sometimes we express the proteins from outside. It is also, the signal can also come from proteins bound with fluorescently labeled antibodies, and we are sometimes using fluorescent dyes for staining of different cell parts like DNA, nucleus, mitochondria, or so many other things. So we are having a list of fluorescent dyes here which can be used to tag the antibodies or molecules. So here is the list of dyes like FITC, PE, APC, PERCP, Alexafluor 488, Alexafluor 647, and Pacific Blue. There are multiple other dyes are present like PERCP 5.5, PE size 7, APC size 7. So many color choices are present there. And here is the list of absorbance and emission of these fluorescent dyes. Other than these fluorescent dyes, the fluorescent thing or the fluorescent signal can come from fluorescent proteins. Here are some examples like CFP, green fluorescent protein, yellow fluorescent protein, and red fluorescent protein. Again, we are having their absorbance and emissions here. So what do you think? How many fluorescent signals can be detected by a flow cytometer? So the answer is that depends upon how many lasers we are using in the instrument. So one laser instrument generally creates 4 color detection system, 2 lasers 8 color detection system, 3 lasers 14 color, and there are so many advanced flow cytometer nowadays which can detect 18 colors at a time. It means FSC size, SSC granularity, and other than that you can analyze 18 proteins at a time. It means you can use 18 fluorescently labeled antibodies, or you can detect 18 different kind of fluorescent wavelength. So this was the basic principle. You arrange the cells in single array, cells will interact with light, can give you information about size, granularity, and fluorescent signals. Now we will be doing one experiment to understand the things which we have discussed till now. So in this experiment, what we are doing? We are taking blood first, and by using a Ficol gradient method, or by lysing the RBCs, from this blood we will purify PBMCs. PBMCs are nothing but peripheral blood mononuclear cells. Now these PBMCs we are going to stain with fluorescently labeled antibodies. It means we will mix the fluorescently labeled antibodies with our PBMC suspension system. And here in this experiment we are using the antibodies against CD3 and CD19, and we are putting the whole sample in flow cytometer. So before going ahead, I just give you information about CD. So what is CD? CD markers are cluster of differentiation markers. These are the surface proteins which are present on specific cell types which can be used for the identification of those cells. For example, the lymphocytes or the T lymphocytes can be divided into two parts or two types of T cells, CD4 lymphocyte and CD8 lymphocyte. Means CD4 molecule, a protein which is present on CD4 cells, and CD8 is a protein which is present on CD8 lymphocytes. So there are total 371 CD markers. So based on these markers you can differentiate, you can identify, you can measure the cell types. I just give you a small example. If we talk about regulatory T cell, then the identification phenotype of the regulatory T cell is like this. Regulatory T cells are CD4 positive, CD25 positive, or CD25 high. It means they have higher expression of CD25. They are FOXP3 positive. They are CD152 positive. Means you can use a group of CD molecules, and by using those CD molecules you can identify or you can characterize a specific cell. So here in this experiment we are using the antibodies against two CD molecules. One is CD3 and one is CD19. So here in this table you can see that CD3 is a protein which is present as surface protein on T lymphocyte, and CD19 is present as surface protein of B lymphocyte. So by using these two proteins I can differentiate between T lymphocytes and B lymphocyte. So once we are putting them in flow cytometer, then which kind of result we are going to get, we just see. So this kind of result we are going to get. So in first graph on x-axis I am having FSC, that is forward scatter, which is going to give me information about size of cell. Now on y-axis I am having SSC, that is side scatter, which is going to give me information about the granularity of cell. Now based on the values of FSC and SSC, the blood cells of PBMCs give us three distinct populations which you can see in these circles, right? So the first circle is giving us the information about lymphocyte. Here in this graph, the graph is called as dot plot, and one dot is indicating one cell. Now the cells which are coming in this circle are lymphocytes, cells which are coming in this circle are monocytes, and cells which are coming in this circle, they are granulocytes. So based on FSC and SSC, you can differentiate that in PBMCs you can have three types of cells: monocyte, lymphocyte, and granulocyte. It's like Microsoft Paint. You can go in the software and you can select the population. So here we have selected the lymphocytes because T cells and B cells are present in the lymphocyte. So we will tell the software we want to analyze the cells which are present in this circle. It means we want to analyze the cells which are lymphocytes. So now these lymphocytes are analyzed here. So on x-axis we are having CD3 and on y-axis we are having CD19. So what is happening here? These CD3 molecule, we have used the antibody against the CD3 molecule, which was stained, or which was the antibody was labeled with elixafluor 647, which is a fluorescent dye, and on y-axis we are having CD19. It means we have used the antibody against CD19, and that antibody was labeled with picorithrine or PE. So based on CD3 and CD19, when we look at the cells, then we find that there are some cells which are CD3 positive here in this circle, and there are some cells which are CD19 positive which are in this circle. So this circle is telling us the percentage of T cells, and in this circle we are getting the percentage of B cell. So in a simple experiment, first based on size and granularity, we got lymphocyte, monocyte and granulocyte, and then based on CD3 expression and CD19 expression, we got the percentage of T cell as well as the percentage of B cell. It is so simple to do. So guys, till now everything is looking fine, everything is looking easy, but here comes the first problem. And the problem is uh, when we say that we can use multiple colors or multiple antibodies to recognize multiple proteins, we face a big challenge, and that challenge is when you select multiple colors, their spectrum can overlap with each other and we can get false result. So each color goes in a specific channel in flow cytometer, but because of spectral overlap in colors, one color can go in more than one channel, or one color can be detected by more than one channel, which can give us false result. So here we will see the graphical explanation of this, and in next slide I will be explaining the practical thing about this problem. So if I see the graph emission absorption of FITC, or if I see the wavelength of PE, then I find an overlapping region. It means FITC is entering in the channel where PE is detected. So we can get false result in this condition. Now in another graph we are having so many colors like FITC, PE, PC5 per CP, and these dyes are giving overlapping spectrums. It means they can also give us false result. Now to solve this problem, there is a mathematical model or mathematical system which is called as compensation. So compensation is a mathematical method to solve the issue of spectral overlap, or the issue which is created by a condition where one color can go in more than one channel. So even if it is difficult to understand by these graphs, we can take a live example, and by looking at the example you will understand what I am trying to explain here. So in this thing we have stained the cells with a marker which is called as CD4, and CD4 was labeled, or anti-CD4 antibody was labeled with FITC, which is a color. Now in this experiment we are using only a single color, that is FITC. But when I make a graph where on x-axis I am having FITC, and on y-axis I am having PE, that even if we didn't add any PE antibody, we are having some cells going in PE channel. So it means the FITC is overlapping with PE, and FITC is also detected by PE channel. That is why we are getting this kind of graph, or these cells which are looking like both FITC positive and they are also looking like PE positive. So when we apply the compensation, this graph converts in this situation where we have only and only FITC positive cells. We don't have any PE positive cells. It is very simple to understand. Now we understand with the help of two colors at a time. So on x-axis we have used CD4 anti-CD4 antibody, which is labeled with FITC, and on y-axis we have used anti-CD19 PE antibody. So what is happening? These are PE positive cells. These are negative for both the colors. These are the cells which should be CD4 positive or FITC positive, but because compensation is absent, these cells are going in this region. It means they are looking like they are FITC positive as well as they are PE positive. But once the compensation is applied, this graph is converting into another situation where we are having only CD4 FITC positive cells. Means now FITC is not going in PE channel. We are not having these kind of diagonal cells. Okay, so compensation is a method. It is easy to do, it is easy to understand, where we are solving the problem which is created by a condition where one color can be detected by more than one channel, right? So practically it takes some time to understand the compensation, or it takes some time to solve the issue, but yeah, with practice you can do anything. Now we go for the applications, and first application is the detection of live and dead cell. So in this experiment what we are doing, we are having some cells. These cells are stained with dyes which are propylium iodide, or you can use another dye, that is 7-AAD. There are some other dyes also like Cytox blue, Hox and other things. Once you stain the cells either with propylium iodide or 7-AAD, you put the sample in flow cytometer. So now we just get some information about PI and 7-AAD. So PI can be detected by the laser of 488 nanometer. Propylium iodide is detected by 488 nanometer laser of flow cytometer, and this dye binds to double-stranded DNA, and this dye cannot enter inside the live cell. It means this dye can only and only go in the dead cells, where it will be binding to double-stranded DNA, it will be intercalated there, and it will be giving a specific fluorescence, and that specific fluorescence is detected by 488 nanometer laser, excited by 488 nanometer. Another dye is 7-AAD. It is again excited by 488 nanometer, doing the same thing, binding to double-stranded DNA and excluded by live cell. It means it can also enter only and only in dead cell. So when we put the sample in flow cytometer and when we analyze, we get this kind of graph, where on y-axis I am having side scatter, and on x-axis I am keeping the fluorescence given by PI positive cells. So here is the thing. So the cells which are right to this thing are PI positive, means they are dead cell, and cells which are left, or cells which are present in in this gate. In flow cytometry, the selection area is called as gate. The cells which are present in this gate are live, those are 87.6%. So just by looking at the fluorescence given by the propylium iodide, you can detect or you can differentiate between live cells and dead. Now we see the second interesting application of flow cytometry, that is analysis of apoptosis. Apoptosis is a pathway where the programmed cell death is occurring. It means apoptosis is one of the cell death pathway, and we can analyze it by using the flow cytometry. So we just understand the basic principle of this application. So we are having live cells or healthy cells. Inside the healthy cells, we are having presence of phosphatidylserine, which is present inside the cell on the plasma membrane. If a cell is entering in apoptosis, then membrane flips and the phosphatidylserine comes out, and we use a fluorescent labeled protein, that is annexine, and annexine is having the capability to bind to the phosphatidylserine, which is now flipped outside. So initially the cells which are alive and healthy, they cannot be bound with annexine, and they cannot be also stained by the propylium iodide, because we have seen that propylium iodide can enter only in the dead cell. So the live and healthy cells are annexine negative as well as PI negative. Once the cells are going in early apoptosis, the phosphatidylserine is coming out, and now you are having annexine positive cells, because annexine can bind to that phosphatidylserine which is present outside of the cell membrane. But still the cells are propydium iodide negative. Why? Because propydium iodide can enter only and only in the dead cell. Finally, in the late apoptosis, the propydium iodide will enter inside the cell and cells will become annexine positive, propydium iodide positive. So we are having three conditions: live and healthy cells, annexine negative, and they will be also Pi negative. Early apoptotic cells, annexine positive but Pi negative, and late apoptotic cells, annexine positive as well as Pi positive. So we do the experiment, and in this experiment we are taking again the cells, staining them annexine 5 and propydium iodide, and we are putting them in the flow cytometer. This graph we are going to get. So on x-axis we are having annexine, we have FITC labeled annexine, and on y-axis we are having propydium iodide fluorescence. So these are the cells which are annexine negative as well as Pi negative. It means they are our healthy and live cells. These are the cells which are only annexine positive but Pi negative. They are early apoptotic cells. These are the cells which are positive for both Pi positive as well as annexine positive, and they are dead because of apoptosis. So this kind of beautiful graph we get, and machine can calculate the percentage of live cell, early apoptotic cells as well as dead cells because of apoptosis. Here I am not mentioning the name of drug, but this is our control experiment. These cells are treated with two different concentration, low concentration as well as high concentration of the drug. So this is the low concentration treatment, and we can see now we are having increased cells in apoptotic and increased dead cells. And when you are increasing the concentration more, then you are getting very high number of apoptotic cells as well as dead cells. So this kind of analysis of apoptosis by using flow cytometry can be used for anti-cancer drugs or cytotoxic drugs, and it is a very important application in pharmaceutical research. So again, healthy cells are negative for both, apoptotic cells are positive for annexine, negative for Pi, dead cells are positive for annexine as well as positive for Pi. It is a very interesting application where we are analyzing the apoptosis by flow cytometer. Now we move to third application, that is analysis of cell cycle.